The next speaker would be Dr. Raissa Gutierrez from Instituto de Física de São Carlos, University of São Paulo, in Brazil. She sent us a video recording from her talk, which I'll start presenting now.
Hi everyone, my name is Raissa and I am a PhD student from University of São Paulo, Brazil. I am pleased of presenting my doctorate research in the Young Scientist webinar series. Today I will present a novel insight into the thiaminase type 2 function of EfTenA protein from Enterococcus faecalis. Thiamine, commonly known as vitamin B1, is an essential vitamin for all living organisms. The diphosphate form of thiamine, TPP, is an important active cofactor to the metabolism of carbohydrates, amino acids, and other organic molecules. TPP is synthesized de novo by bacteria, archaea, yeast, fungi, plants, and protozoans. In all cases, thiazole and pyrimidine precursors are synthesized in separated groups and then coupled to form thiamine phosphate, TMP. Then a final phosphorylation step gives TPP, the active form of the cofactor. Over the past 20 years, biochemical and structural studies have elucidated most of the details of the thiamine biosynthetic pathway in prokaryotes and yeast, but there is still much to discover about this pathway regarding the thiamine metabolism, regulation, transport, and salvage in other microorganisms. I will refer to the thiamine biosynthetic pathway as TBS for easy comprehension. But why is the importance of better understanding TBS in other microorganisms? It has been a while that the World Health Organization warns that antimicrobial resistance is a big threat to public health. As the existing antibiotics are limited to 5 general targets, such as cell wall biosynthesis, protein synthesis, DNA replication and transcription to RNA, folate biosynthesis, and the disruption of the membrane, the discovery of new targets and antibiotics is urgent. So, in this context, the TBS pathway in pathogens has been proposed as a potential source of drug targets for the development of new antibiotics, since mammals cannot synthesize TPP de novo and depend exclusively on the dietary uptake of thiamine. Here are some examples of recent efforts of validating TBS pathway as a source of targets for human pathogens. The thiamine metabolism is complex and involves the biosynthesis of precursors, regulation at transcriptional level, transporting, salvaging, and recycling. The HMP and THZ biosynthesis is the most variable part of the TBS pathway, and the synthesis of TMP involving Thi-D, Thi-E, and Thi-M enzymes is the most conserved part. So, these 3 enzymes are good candidates for antibiotic development. In addition, 2 types of thiamine-degrading enzymes are called thiaminase 1 and 2, have been characterized and they are recently linked to ATMP salvage pathway. Although their reaction mechanism is similar, the 2 proteins lack sequence similarity. At the first impression, it sounds paradoxical that the TBS pathway has a thiamine degradative enzyme. Here, we are interested in the thiaminase 2, known as ThenA, that is widely distributed in bacteria and archaea, with homologs also found in fungi and plants. ThenA is intracellular and it uses water as a nucleophile for the base exchange reaction. ThenA also has an amino hydrolase activity and can recycle base-degraded thiamine that is important by the cell, restoring HMP, which can be used in the thiamine synthesis. TMP and TPP are not described as ThenA substrates, but the yeast homolog can hydrolase thiamine antagonists as pyrithiamine and oxythiamine. Despite all advances, the biological function of thiaminases is not completely understood, so it has been also speculated that the thiaminase could play an important role not only in the salvage of thiamine precursors, but also in detoxifying and recycling components of thiamine anodes. In this work, we propose to study the TBS pathway from the pathogen Enterococcus faecalis, a gram-positive commensal bacterium present in the normal flora of the human gastrointestinal tract and associated with hospital infections. The last CDC report treats vancomycin-resistant Enterococcus strains as a serious threat once these strains are expanding resistance to additional antibiotics, leaving few or no treatment options for patients. A blast search in the faecalis genome identified thiamine biosynthetic genes, suggesting this human pathogen can synthesize thiamine de novo, although the complete set of biosynthetic genes for the precursors was not identified. Transporters for thiamine, THZ, and HMP are predicted as well. In faecalis, the THA gene is under TPP riboswitch regulation, and it is clustered with an HMP transporter, suggesting it may play a role in HMP salvage. So we want to investigate EfTenA activity against thiamine, TMP, and TPP, and evaluate if the thiaminase activity is relevant as a degradative enzyme. We amplified the TNA sequence code from a genomic DNA library and cloned it into an expression vector. After purification steps and initial biophysical characterization, we used the protein purified to carry out binding affinity assays, activity assays, and crystallization screening. The determined structure was used to evaluate the binding mode of thiamine and its phosphorylated forms through docking simulations. EfTenA protein has 220 residues and 25 kilodalton. The recombinant protein is extremely soluble and stable, and its oligomeric state as a dimer in solution was confirmed by size exclusion chromatography and by analytical ultracentrifugation. Also, a circular dichroism spectrum in tris or phosphate buffer shows that EfTenA is folded as an alpha helical protein with no major structural changes upon buffer exchange. In order to investigate the EfTenA interaction with thiamine and its phosphorylated forms, a thermal denaturation experiment was set up in the presence of increasing concentration of the three compounds. It is interesting to note that TMP and TPP are not described as TNA substrate for other homologs, but the TSF experiment showed that EfTenA melting temperature increases in the presence of these three compounds. So this qualitative result clearly indicates that the phosphorylated thiamine species can interact with EfTenA, with decreasing binding affinities as the phosphate groups are added to the thiamine. So the data suggests that the addition of phosphate groups has a negative effect on affinity, probably by steric hindrance or charge repulsion in the vicinity of the active site. Next, the thiaminase activity was evaluated by measuring the decrease of the substrate concentration over time using thiochroma assay. We found that EfTenA is active on thiamine, with a specific activity of about 28 nanomole per minute milligram. It's also poorly active on TMP, with a specific activity of 0.7 nanomole per minute milligram, and we considered the EfTenA is inactive on TPP. In comparison with TNA from S. aureus and Bacillus subtilis, EfTenA has a moderate thiaminase activity, but is the only one which has unless a poor activity on TMP. We also determined the EfTenA crystallographic structure at 1.9 angstroms. We used the TNA homolog from Bacillus subtilis that shares 40% in sequence identity with EfTenA as a model for molecular replacement. In agreement with the species observed in solution, EfTenA was found as a dimer in the asymmetric unit. Each monomer is composed of 11 alpha helices and 3 PEG molecules were modeled in the interface between them. Also, an imidazole molecule that was present in the crystallization condition was observed bound into the active site of each monomer. In an attempt to obtain a structure of the complex EfTenA-TMP, we added TMP to the EfTenA crystallization trial after crystals stopped growing. The novel crystal structure, determined at 2.2 angstroms, revealed the presence of HMP in the active site instead of TMP. The HMP product interacts with EfTenA by 2 hydrogen bonds with the aspartic acid 44 and 1 hydrogen bond with the glutamic acid 205. The presence of the HMP product in the active site after catalysis supports a possible inhibition by the product, corroborating the findings that thiaminase reaction from other homologs does not follow a nuclease-maintaining kinetics. Docking calculations using Libella program was used to evaluate the most probable binding mode for thiamine, TMP, and TPP in the active site. In the docking pose for thiamine, the THZ ring is docked in the pocket between serine 136 and tyrosine 163. The terminal hydroxyl group in thiamine is solvent exposed and was not found favorable interactions with any protein polar group. The energy computed for this pose was minus 36 kcal per mole, indicating a reasonable interaction. For TMP, similar conformations was found in docking calculations, and the phosphate group is somewhat stabilized by favorable interactions with histidine 171. On the other hand, the carboxylate group of glutamic acid 167 is found at 5.4 angstroms distance from the phosphate group. Since electrostatic energy are long-range terms in the force field calculations, the interaction energy becomes less favorable by the introduction of the phosphate group in TMP as compared to thiamine, and the overall energy computed for TMP was minus 7.5 kcal per mole. The electrostatic contribution for the interactions between EfTenA and TMP was of minus 5.3 kcal per mole and minus 16.4 kcal per mole for thiamine. It was not possible to obtain a low-energy binding pose for TPP, and we attribute the failure to the excessive repulsive interaction between the pyrophosphate group and glutamic acid 167. To conclude, based on the genomic search, it is unlikely that intracellular TNA exerts a degradative activity on thiamine, TMP, or TPP. First, because TNA gene is found under regulation of the TPP ribo switch, and second, because faecalis has roots to salvage and use the precursors and thiamine glutamines. If faecalis uptakes the THZ and HMP precursors, they probably are directed to the de novo pathway. If faecalis uptakes thiamine, it could be pyrophosphorylated to TPP by TPK. And if faecalis uptakes TMP, it could be dephosphorylated by a GTPase to thiamine and then pyrophosphorylate to TPP. And if faecalis uptakes TPP, it is ready for use. Briefly, we found that TNA is moderated active on thiamine, poorly active on TMP, and inactive on TPP. HMP product remains bound to EfTenA after TMP hydrolysis. and the decreasing affinity in the interaction between EfTenA with thiamine, TMP, and TPP is explained by the negatively electrostatic effect in the interaction of the phosphate groups with the vicinity of EfTenA active site. So, taken together, the genomic analysis and our results suggest that thiaminase activity as a degradative enzyme is probably not the biological function for 10A enzyme in Enterococcus faecalis. So, our perspectives for the future are to evaluate the nutritional requirements of Enterococcus faecalis regarding precursors and thiamine vitamins. Also, we want to investigate EfTenA participation in the AGMP salvage pathway from thiamine analogs. I want to thank the IUPAB-SBBf-SBBq organizing committee for the opportunity, the facilities research that I used in this project, the sponsors, and my research group. Also, I want to thank you that are watching my presentation. If you have any questions or suggestions, do not hesitate to contact me on my email. Thank you.